Bismillah ar-Rahman ar-Rahim. We follow up together the endocrine system, and we have finished the suprarenal cortex, and the lecture of today is the suprarenal medulla part of suprarenal gland and pineal gland. By the end of this lecture, you should be able to describe the structure, which includes the site, light and electron microscopic picture of the suprarenal medulla cells, and describe the structure, site and light and electron microscopic picture of the pineal body, and correlate the structure of the above glands, suprarenal medulla and pineal body, with their function. How the cells of these two glands adapted in structure to perform their function. In the previous lecture, we studied that suprarenal gland parenchyma formed of cortex and medulla, outer cortex and inner medulla. The lecture of today is the suprarenal medulla. And as we mentioned, suprarenal cortex formed of three zones: zona glomerulosa, zona fasciculata, and zona reticularis. Today, we will study suprarenal medulla. Suprarenal medulla is different in origin from the suprarenal cortex. Suprarenal cortex originated from mesoderm, but suprarenal medulla originated from the neural crest. Suprarenal medulla appears in fresh state, reddish in color. Reddish in color as it is highly vascular. Suprarenal medulla highly vascular, so it appears in fresh state, reddish in color. It's formed of two types of cells. This is the suprarenal medulla. It's formed of two types of cells: chromaffin cells and ganglion cells. Chromaffin cells and ganglion cells. Chromaffin cells are the main cells in the suprarenal medulla. They are modified sympathetic post-ganglionic cells. Why it's called modified, and why it's called sympathetic, and why it's called post-ganglionic? It's called modified sympathetic as it is lack axons and dendrites, and become secretory cells to secrete epinephrine and norepinephrine. It's not as any nerve cell. It's lack axons and dendrites and become secretory cells. So it's called modified sympathetic. Called the post-ganglionic cells as they are innervated by preganglionic sympathetic nerve fibers. So they are considered as post-ganglionic cells, as they are innervated by preganglionic sympathetic nerve fibers, so they called post-ganglionic cells. This slide microscopic picture to the suprarenal medulla. This is the chromaffin cells. The chromaffin cells, and between chromaffin cells we will find fenestrated blood sinusoids or blood capillaries, as any endocrine cells to take the secretion directly to the blood circulation in them. Chromaffin cells. Chromaffin cells are the main type of cells in the suprarenal medulla. This slide microscopic picture to suprarenal medulla showing the chromaffin cells which appear with basophilic cytoplasm and large vesicular eccentric nucleus, as these cells active in secretion of adrenaline and noradrenaline, or epinephrine and norepinephrine. So these cells appear by light microscope with basophilic cytoplasm and large vesicular eccentric nucleus. And these groups of cells, which are polyhedral cells, these groups of cells separated by fenestrated sinusoidal capillaries, capillaries to take the secretion of these cells directly to the blood circulation. These cells contain in its cytoplasm, the cell contain in its cytoplasm fine granules showing certain staining characters, as when we stain the suprarenal medulla or suprarenal gland by chromium salts, this part or suprarenal medulla give positive chromaffin reaction, as these chromaffin cells stained positive by chromium salts. So it's called chromaffin cell. By electron microscope, chromaffin cells shows the picture of protein secreting cells. What is the picture of protein secreting cells? Protein secreting cells must contain well-developed rough endoplasmic reticulum that synthesize protein and well-developed Golgi apparatus that add carbohydrate to protein and package the protein, and mitochondria that give energy to this process, and membrane-bounded granules that secreted from the Golgi apparatus and contain the secretory product inside. So, as any protein synthesizing cells, chromaffin cells by electron microscope contain well-developed rough endoplasmic reticulum, well-developed Golgi apparatus, mitochondria, and membrane-bounded granules. At ultrastructural level, chromaffin cells are two types according to the product or the secreting substance. The first type, epinephrine secreting cells, and the second type, norepinephrine secreting cells. Epinephrine secreting cells is different from the other cells according to the granules inside. By ultrastructural level, the granules that contain epinephrine are homogeneous granules and less electron dense. Homogeneous granules and less electron dense. But norepinephrine secreting cells contain granules that have electron dense core with light halo around. Electron dense core with light halo around. So there are two types of cells or two types of chromaffin cells at ultrastructural level. Epinephrine secreting cells contain granules homogeneous and less electron dense, and norepinephrine secreting cells, the granules are electron dense core and light halo. We mentioned that by electron microscope or at ultrastructural level, we have two types of chromaffin cells, epinephrine secreting cells and norepinephrine secreting cells. This is electron microscopic picture to norepinephrine secreting cells. And any chromaffin cells, whatever the norepinephrine or epinephrine secreting cells, take the characters of protein secreting cell, as it contains well-developed rough endoplasmic reticulum, well-developed Golgi apparatus, many mitochondria. So it takes the character of protein synthesizing cell. But the granules inside their cytoplasm show that these cells are norepinephrine secreting cells, as these granules contain electron dense core surrounded by peripheral clear halo. Electron dense core surrounded by peripheral clear halo. This is the characters of norepinephrine secreting cell, which is a type of chromaffin cells. As we mentioned, the suprarenal gland or adrenal gland formed of two parts cortex and medulla. The medulla formed of two types of cells, the chromaffin cells and ganglion cells. Why the chromaffin cells called by this name? This is due to the staining methods of the adrenal gland. When we stain the adrenal gland, all the adrenal gland by chromium salts is potassium dichromate. The adrenal medulla part shows positive chromaffin reaction. When we stain adrenal gland by chromium salts, the only the medulla part which take the chromium salts and give brown color or positive chromaffin reaction, as it contains catecholamines, which are adrenaline and noradrenaline. This is the cause that these cells are called chromaffin cells, as it gives a positive chromaffin reaction when the adrenal gland stained by chromium salts, and the cortex part gives a negative reaction, the positive reaction to give brown color. Another test called the Cramer's test. Cramer's test. What is this test? This test by two steps. The first step, we can treat the adrenal gland by osmic acid, and the second test is to treat adrenal gland by turpentine oil or turpentine oil. When treating first section of the gland with osmic acid, as you studied before, osmic acid stained the tissue with black color. Stained the tissue with black color when there is fat or catecholamine in the tissue. The cortex contains fat, as all cells of the cortex secrete steroid hormones, and the medulla contains catecholamines, adrenaline and noradrenaline. The two parts take the osmic acid and give black color, so all the gland, cortex and medulla stained black by osmic acid. How we can differentiate between cortex and medulla? By treating the gland after osmic acid by turpentine oil. Turpentine oil dissolves the fat in the cortex. So the cortex will be white and medulla is still black. So we can differentiate by Cramer's test between the cortex and the medulla of suprarenal gland, and also by chromaffin reaction we can differentiate between cortex and medulla, as the medulla give positive chromaffin reaction as it stained brown. It stained brown when we stain all the gland with chromic acid as potassium dichromate. Ganglion cells are the second type of cells present in the adrenal medulla. As we studied, adrenal medulla formed of two types of cells. All cells in adrenal medulla are sympathetic ganglion cells. Some of these cells, or many of these cells, the main cells, chromaffin cells, are modified sympathetic cells that loses their axons and dendrites and become secretory to secrete adrenaline and noradrenaline, or epinephrine and norepinephrine. These cells form the main type of cells in the adrenal medulla. The second type, which is few cells, are called ganglion cells. These cells are not modified. They are sympathetic ganglion cells that stimulate secretory activity of the chromaffin cells. The ganglion cells are star-shaped cells with basophilic cytoplasm, as you see, with basophilic cytoplasm and the vesicular excentric nucleus. So the adrenal medulla formed of two types of cells. Chromaffin cells are the main type of cells, and ganglion cells which are few cells scattered between the chromaffin cells. This diagram to illustrate the suprarenal gland or the structure of suprarenal or adrenal gland. Adrenal gland formed of stroma and parenchyma. Adrenal gland formed of stroma and parenchyma. The stroma formed of capsule which surround the gland and separate it from surrounding structure. This capsule formed of fibroelastic connective tissue. From the structure of capsule, trabeculae or stroma descend to divide the suprarenal cortex into compartments. Then the parenchyma, which contains the functioning cells that secrete hormones, formed of cortex and medulla. The parenchyma of suprarenal gland formed of cortex and medulla. The cortex are the peripheral part. The cortex, the peripheral part which lies directly under the capsule. And the parenchyma is the inner central part of the gland. The cortex formed of 3 zones. The first zone, which lies directly under the capsule, called zona glomerulosa. Zona glomerulosa. As the cells of this zone are columnar cells and arranged in glomeruli. These glomeruli separated by fenestrated blood capillaries. Fenestrated blood capillaries. As these cells secrete aldosterone hormone, which regulates the electrolyte and water balance under the effect of renin enzyme. The second zone called zona fasciculata. Zona fasciculata. This zone formed of cords of cells. Cords or columns of cells beside each other and separated by fenestrated blood capillaries. Fenestrated blood capillaries. This zone secretes the main hormone of suprarenal gland, which is called cortisol. Cortisol hormone is an anti-inflammatory and immunosuppressive hormone. These cells of the zona fasciculata called spongiocytes. Spongiocytes. As these cells have vacuolated cytoplasm. Contain many fat droplets, so the cytoplasm appears vacuolated or sponge-like. So it's called spongiocytes. Third zone called the zona reticularis. Zona reticularis, as it forms of a reticulum, as the cords of cells are branching and anastomosing cords. These cells separated also by fenestrated blood capillaries, as they secrete androgen. As these cells secrete androgen directly in these fenestrated blood capillaries. Then the second part of the parenchyma called the medulla. The medulla, as we studied, formed of 2 types of cells. The main type called chromaffin cells, and called chromaffin as it gives positive result when we stain the suprarenal gland by chromium salts. This part stained brown and give positive result as these cells secrete catecholamines, epinephrine and norepinephrine, which give positive results with chromium salts. The second type are the ganglion cells. These ganglion cells appear basophilic cytoplasm and vesicular eccentric nucleus, and these cells are sympathetic ganglion cells to stimulate the activity or secretion of chromaffin cells in the suprarenal medulla. And also the cells of the medulla separated by fenestrated blood capillaries to secrete catecholamines directly in these capillaries to reach the blood circulation. Paraganglia. Paraganglia are collection of catecholamine secreting cells adjacent to the autonomic ganglia. So it's called the para as these cells adjacent to the autonomic ganglia. The type of catecholamine secreting cells which present in paraganglia are norepinephrine secreting cells. So these cells give positive chromaffin reaction also with the chromaffin cells in adrenal medulla. So we mentioned paraganglia in this lecture as it gives positive chromaffin reaction as the chromaffin cells in adrenal medulla. The conversion of norepinephrine to epinephrine occur only in the adrenal medulla. Pheochromocytoma. Pheochromocytoma is a benign tumor of the adrenal medulla. So there is excretion of high level of catecholamines, adrenaline and noradrenaline, which gives sympathetic stimulation. So the symptoms of this pheochromocytoma are high blood pressure, headache, heavy sweating, rapid heartbeat, tremors, pallor, all symptoms of sympathetic stimulation. If this pheochromocytoma isn't treated, severe or life-threatening damage to other body systems can result. So this pheochromocytoma removed surgically to return the blood pressure and all symptoms to normal. These are two light microscopic pictures to the suprarenal gland or adrenal gland, showing that suprarenal gland covered by capsule. When we refer to this part by arrow, this is the capsule and this is the trabeculae. Then we start in parenchyma, the zones of the cortex of the parenchyma, zona glomerulosa, zona reticularis fasciculata, zona reticularis. Again, zona glomerulosa, zona fasciculata, zona reticularis. And this part called the medulla. All the cells in the suprarenal gland, parenchyma, whatever cortex or medulla, are separated by fenestrated blood capillaries. Fenestrated blood capillaries to secrete hormones directly to the blood circulation. This is also light microscopic picture to the suprarenal gland or adrenal gland, showing that the gland covered by capsule, and this part zona glomerulosa, and this zona fasciculata, zona reticularis, and the medulla. Now we have finished the suprarenal gland and we will start in pineal body or pineal gland. Pineal gland is a cone-shaped gland attached to the base of the brain. We have two glands attached to the base of the brain, pituitary gland situated anteriorly and pineal gland situated posteriorly and at higher levels than the pituitary gland. Pineal gland originated from neuroectoderm. So pineal gland developed from neural ectoderm and it is a cone-shaped gland attached to the base of the brain. However, pineal gland lies in the brain, but they formed histologically as any parenchymatous organ, formed of stroma and parenchyma. The stroma in the pineal gland formed of thin capsule, but the capsule not formed of connective tissue. It's formed of biomatter as it located in the brain. Thin capsule of biomatter and also thin connective tissue septa containing blood vessels and nerve fibers descend from the capsule to divide the pineal gland into incomplete lobules. And reticular network that form the background of the gland and support the gland. Then the parenchyma, which contains the functioning cells The parenchyma formed of two types of cells, pinealocytes and neuroglial cells. Pinealocytes are the main cells of the parenchyma and are the functioning and secretory cells in the parenchyma. And the neuroglial cells, as we studied before, to support, it is the support in the brain, support cells in the brain. So these neuroglial cells support the functioning cells, pinealocytes. This is a light microscopic picture to the pineal gland, showing that pinealocytes by light microscope, arranged in groups, are large cells and arranged in groups beside each other. The nucleus of these cells appears large irregular nucleus, achromatic with apparent nucleolus, as these cells are very active cells in secretion, so its nucleus appears achromatic with apparent nucleoli. The cytoplasm of cells is pale, slightly basophilic with few lipid droplets. This diagram showing the two types of cells present in pineal gland. The first cell is the pinealocytes, and the second type neuroglial cells which support pinealocytes. The main type pinealocytes are large cells with long branching processes. These processes not apparent in hematoxylin and eosin stent section. So we make impregnation with silver salts to show these long branching processes. These branching processes end in dilatation, which make contact with the blood capillary, which take the secretion of the gland to secrete it in the blood capillaries. These cells are very active cells, so the cytoplasm is pale basophilic and the nucleus is large vesicular nucleus with apparent nucleoli. And this is the neuroglial or support cells to the pinealocytes. This diagram showing the electron microscopic picture of the pinealocytes, as it is large cells with cytoplasmic processes. By electron microscope, these cells formed of many smooth endoplasmic reticulum, numerous mitochondria, few rough endoplasmic reticulum and Golgi apparatus, and electron dense granules, and the cell processes. To give the shape of the cell processes contain microtubules. Microtubule give the processes its shape or their shape. And the small vesicles at the end of cytoplasmic processes. These processes make contact with the blood capillaries. So this is the electron microscopic picture of pinealocytes, which are the main cells of pineal gland. As we saw in the previous diagram, neuroglial cells, which are modified astrocytes, lies between the groups of pinealocytes. The neuroglial cells lies between groups of pinealocytes. They are few cells with deeply stained nucleus. as these cells not active secretory cells, so they have a deeply stained nucleus. These cells are triangular in shape with long processes as any nerve cell, triangular with long processes. The function of these cells are supporting cells and forming network around the pinealocytes to support the pinealocytes. Another structure appear in aging pineal gland. As age causes extracellular protein deposition and formation of calcified bodies called brain sand or concretion. These brain sand or these calcified bodies are calcified secretory products accumulated in the form of concentric layers to form corpora arnica. This is like microscopic picture to young pineal gland, and this is to aging pineal gland. Brain sand or corpora arnica appears in aging pineal gland. This structure appears clearly by X-ray. By X-ray appears clearly and considered as a landmark to localize any mass in the brain. If there is any mass in the brain, will displace the position of this corpora arnica. So it's considered as a landmark for any mass in the brain which appear by X-ray. Control of pineal gland. Pinealocytes, which are responsible for the main secretion of the pineal gland, innervated by post-ganglionic sympathetic fibers. These fibers are unmyelinated nerve fibers, which synapse with pinealocytes. At the end of these fibers, there is synaptic vesicles. At end of each fiber, there is synaptic vesicle. Synaptic vesicle contain noradrenaline, which stimulate pinealocytes. Pinealocytes release two secretions, serotonin during daylight and melatonin during night. So the pinealocytes or pineal gland responsible for diurnal rhythm or sleep rhythm, as it secretes serotonin during daylight and melatonin during night, which gives a sleep rhythm or diurnal rhythm. It also suppresses gonadotropin secretion. This gonadotropin prevent the growth and maturation of gonads until puberty. The pineal gland suppresses this secretion, which consequently prevent the growth and maturation of gonads until puberty. So the pineal gland is large during infancy and involutes after puberty. Large during infancy to suppress the gonadotropin secretion and suppress the maturation of gonads until puberty, and then involutes to smaller size after puberty. So it controls the age of maturity, the age of sex maturity. Again, the pineal gland responsible for diurnal rhythm and controls the age of sex maturity. Diurnal rhythm as it secretes serotonin during day and melatonin during night. So it controls the sleep hours. It also controls the age of sex maturity as it suppresses the gonadotropin secretion, which prevent the growth and maturation of gonads until puberty. Paraganglionic or chromaffin system. These systems are a scattered group of cells They are scattered group of cells similar to that of adrenal medulla. As we studied before, adrenal medulla formed of chromaffin cells and ganglion cells. So this system also a group of cells but present at different sites in the body but similar in structure to adrenal medulla. This group of cells, each group of cells, is capsulated and highly vascular as these cells secrete adrenaline and noradrenaline, so they are highly vascular and capsulated to support and protect themselves from surrounding structure. Sites of this system, the first one is the sympathetic paraganglia. We studied it, the paraganglia or sympathetic paraganglia lies beside the sympathetic ganglia and they have the same structure as the adrenal medulla. And also carotid body related to the common carotid artery, and aortic body related to the aorta, and retroperitoneal tissues. All these structures are called the chromaffin system, as they are similar to the adrenal medulla. And when we stain these structures by chromium salt, they give brown color or positive chromaffin reaction. So, it's called the chromaffin system. All these structures are composed of two types of cells, the chromaffin cells and supporting cells. Chromaffin cells and supporting cells. And this is to remember the electron microscopic structure to chromaffin cells, which containing secretory granules that contain epinephrine and norepinephrine. Quiz on the lecture. Brain sand, which is called the corpora arnica, is due to calcium deposition in. As we studied, brain sand is calcium concretion increase with age. This is due to deposition of the secretion or deposition of calcium on the secretory products, which accumulated in concentric layers in the pineal gland. It appears with age, with increased age. So brain sand is due to calcium deposition in the letter C, pineal gland. An example of a mixed endocrine gland is, also we studied in the first lecture of endocrine system that we have pure endocrine gland as suprarenal, parathyroid, thyroid, and mixed endocrine gland. Mixed gland means that part of the gland is endocrine and part exocrine. This is the pancreas, which we will study in the next lecture. The right answer, B, pancreas. A middle-aged female presented with truncal obesity and muscle wasting of limbs. Investigation showed high serum cortisol level. This is due to hyperfunction of. High serum cortisol level means there is high stimulation to the zona fasciculata in the adrenal cortex, which secrete cortisol. The cells in the pituitary that are responsible for stimulation of zona fasciculata are corticotrophs. Corticotrophs secrete adrenocorticotropic hormone, which stimulates zona fasciculata in the adrenal cortex or suprarenal cortex to secrete cortisol. High cortisol level causes truncal obesity and muscle wasting of limbs. So the right answer, letter C, corticotrophs. A hormone secreting cells that shows diurnal variation is diurnal variation caused by serotonin and melatonin. These two secretions secreted from pinealocytes in the pineal gland or pineal body. which are responsible for sleep rhythm or diurnal variation by their secretion, serotonin and melatonin. So the right answer, letter C, pinealocytes, is the hormone secreting cell that shows diurnal variation. Now we have finished the lecture. Thank you. Assalamu alaikum wa rahmatullahi wa barakatuh.